Dr. Peter Marinkovich, who will discuss gene therapy and EB. Dr. Marinkovich does research training in the laboratory of Dr. Robert Bergeson, who discovered type 7 collagen. Dr. Marinkovich's early work led to the characterization of several key basement membrane components involved in EB. He later joined the faculty at Stanford University and served as an attending in the National EB Registry at Stanford University and currently directs the Stanford Blistering Disease Clinic. Dr. Marinkovich's NIH-funded laboratory has had a long-standing focus on the development of molecular therapy for various subtypes of EB. We thank you, and the floor is yours, Dr. Marinkovich.
Well, thank you for the introduction, and thank you everybody for attending today. And I'm just really honored and pleased to be able to talk to all of you. I enjoyed talking at the scientific conferences, but talking to the patients, it's really a special treat. It's inspiring to me to talk to you all, and I feel like I work for Stanford. I get paid by Stanford. I get funded through my salary for some of these companies that I work with as well to do these different therapies. But I really am working for you guys, and so I'm really pleased to be able to this year be able to present something positive for you guys in the field. So I want to just start at the beginning and be able to just give you a little idea as to what we're tackling here. So this is the basement membrane, and so these are the building blocks and the molecular glue that holds the skin together. And so they're like links on a chain. If you have a genetic mutation like Dr. Yudo talked about earlier, of any of these genes coding for any of these proteins, you'll get blistering, and you'll get it at different levels depending on where the gene is mutated. So we'll be focusing mainly on type 7 collagen 7. We'll also touch a little bit on laminin 332, and these two proteins connect to each other in the basement membrane. So first off, let's talk about collagen 7, and that is the protein that is absent in dystrophic epidermolysis bullosa. The split is at the lower lamina densa, so it's at the bottom of the basement membrane. And when collagen 7 is defective, the skin just falls apart in this area due to mechanical trauma. So the goal here is to try to replace collagen 7, and that's what these studies that I will describe, several of them are doing, is trying to put back normal collagen 7 where there's either none or there's a form that's not working. This is what type 7 collagen looks like. It's a trimer. It's sort of, it winds together like three strands of a rope, and it functions like a rope in kind of holding the skin down onto the dermis. It assembles into these structures that are called anchoring fibrils. So you hear me talk about anchoring fibrils, you hear people talk about anchoring fibrils. They're basically just conglomerates, assembled conglomerates of type 7 collagen. So when you don't have type 7 collagen in your skin, as many of you know, you get blistering on your skin. You get a lot of scarring too. And so the severe forms of dystrophic EB, you get bad scarring, wounds that don't heal. You get like this mitten hand deformity, which is, obviously very debilitating. And so it's all of a consequence of the lack of type 7 collagen. You can get squamous cell carcinoma. You can get strictures of the esophagus. So it's a really challenging disease, and so far we really don't have great therapies for it. As families and patients, it's frustrating. It's also frustrating for the physicians because we'd like to be able to do more, but we can provide good wound care, infection, nutrition, treat anemia like Dr. Lucky talked about earlier. Try to find the squamous cell carcinoma, but up until now, we haven't had ways to actively treat the disease. It's been a long road. EB was discovered about over 100 years ago, 1875, And then over time, we've identified the clinical variants. We've been able to characterize the genes and the proteins that are absent in the different subtypes of EB, and that has led to where we are today. So it's just right now we're at the point in a long road and a long journey. So this is a study that came out in 2016 where we did a gene therapy on dystrophic EB patients, where we transferred type 7 collagen to the skin of these patients, the gene coding for type 7 collagen, and then studied them over time. And this was developed at Stanford. I'm but one member of a really talented team of individuals over at Stanford. And together we were able to devise this system where we biopsy the patient, we manufacture the skin cells, we grow them up. Now we're doing it in a GMP facility, which is a good manufacturing facility that's suitable for clinical use. We put the gene back into the cells through a viral vector called a retrovirus. Then we arrange these into sheets that then we allow to mature into sheets about the size of a playing card. And then in the operating room, we place these onto the patient. Patient stays in the hospital for a week to be able to get the grafts to take well. And then we follow the patients over time, biopsying the skin and looking to see whether we're able to correct the skin defect of collagen 7 and also look for wound healing. This is what those grafts look like. They're semi-clear. We place them on top of petroleum gauze, and then we staple them together, put them in this box, and take them into the OR for placement on the patients. And this is done under general anesthesia. These are the first 4 patients we've treated. We've treated actually 3 patients since then for a total of 7. And they all have severe RDEB, recessive dystrophic. And they have no circulating antibodies prior to the grafting, although there's one patient that had some antibodies, and I'll talk about that in a second, that developed during the trial. And so what we found was after we placed the graft, we're able to show some improvement in wound healing. And these are, in some cases, chronic wounds that have been present for many years. So the patients were very happy. We tried to go after the places where the patients thought they would be most useful, and so we tried to work with the patients on this. Here's a few more examples of these grafts that healed over time. This is the shoulder of a patient who had a wound for many years, was never able to sleep on his side, and was finally able to do that after this therapy was implemented. And this shows the clinical responses of the grafts. They did really well initially, and some of them continue to do very well over time. A few of them, some of them started to do a little bit less well. The green is greater than 75% healing, the yellow is 50 to 75, and the red is below 50. So basically, it worked very well over time. And there was some variability, though, and some of the grafts dropped off over time. And we're investigating the reasons why these grafts dropped off over time. But we're very happy to have had these initial positive results. What the gene therapy did was it restored collagen 7 expression. That's this green line you see right here in these micrographs of sections of patient skin from the biopsies. You see that some patients continue to maintain really good expression even through the 6 months, whereas other patients showed, a little bit less expression. Like at 3 months, there's good expression, but at 6 months, there's not. So you can also see in some instances the collagen 7 actually being expressed in the epidermis, these little dots, little green dots. And then what's really cool is being able to show at the super high magnification electron microscopy level the actual localization of the collagen 7 that we're giving these patients to the anchoring fibrils at the electron microscope level. These little dots here represent collagen 7, therapeutic collagen 7, and these anchoring fibrils are coming out of the basement membrane, winding around the collagen fibrils and sort of sewing the basement membrane onto the skin. So an important part of an early phase trial, as Dr. Bruckner had mentioned, is safety. And so we followed the safety very closely. And in general, the patients tolerated the grafts very well. There was no inflammation or other issues such as squamous cell carcinoma. We also, because we were using a virus, we wanted to do safety testing on the virus, and that tested out okay. One thing we did know in one patient is this one patient developed antibodies after transfer of the collagen 7 gene therapy. And this patient, we were actually able to show antibodies in the graft itself on biopsy. So aside from the antibody formation, the patient did well. But this just illustrates an important point for all the different types of molecular therapies that we're doing, is that when you're putting a gene in and a protein presenting to the immune system that maybe the immune system has never seen before, you run the risk of developing an immune reaction. So it's something we need to just follow very closely. And as we move along, this will be an important issue in helping to optimize our gene therapy efforts. So in summary, we've completed this phase 1/2 trial, and we've now treated 7 patients, 6 grafts per patient. We've been able to show long-term collagen 7 expression. Safety was very, very well tolerated. We're still following these patients, and we're gearing up for phase 3 studies. And we've recently partnered with this company, Abeona Therapeutics, who is helping us to manufacture and get the phase 3 trial underway. And for more information on this trial, I would recommend speaking with Melissa Barriga at this number and this email address, and she can tell you more about this trial. So what are some of the lessons that we've learned so far from this grafting study? One thing we know is that it's really important to photograph the wounds very carefully. In order to be able to monitor the wounds, we need to track them with digital photography. And we found that these wounds often will move around. So often they're in the same general location, but they can sometimes heal in one part and extend in another part. And it's sort of like a moving target. So we're appreciating more and more that we need to understand and appreciate the dynamic quality of these wounds as we study their healing. Another thing that we're working hard at trying to understand is the role of the stem cells and the targeting of the stem cells in this type of a cell-based therapy. The fact that we're getting expression for many months or even over a year suggests that we are targeting the stem cells. But what we want to do is we want to target the stem cells as efficiently as we can. And one way to be able to understand that is to do what we call a holoclone analysis, where we take the cells from the patient, expand them into this type of assays that look for progenitor colonies of long-lived cells that give rise to these little dots that you see on the Petri dish. And we have to also understand distinguish the cells that we put on the patient, the transduced cells, versus the cells that are already on the patient. And then we mix them together, so we have to be able to sort out which ones are treated, which ones aren't treated, in order to be able to understand or quantify the efficacy of our therapies. So this is an important ongoing issue we're looking at as well. And then, as I mentioned before, we really have to appreciate and better understand any kind of immune reactivity we see from these types of molecular therapies, and to know when the antibodies are being formed, what part of the type 7 collagen molecule the antibodies are directed against. All this is very important information for us to be able to improve the therapy. I'd like to just take a quick hiatus into junction leprotomyces bullosa. This is another major type of EB that's caused by a split in the center of the basement membrane called the lamina lucida, and involves, in most cases, laminin 332. Laminin 332 is a really interesting molecule. It is the major adhesive protein for keratinocytes. They leave trails of it. That's what you see here in red. They leave trails of it as they move behind, like slime from a slug, as they migrate across the culture surface. It looks like it has a cross shape, and it plays a key role in maintaining adhesion in the skin. There was a study that was published about a year ago in Nature by an Italian group, and in combination with a German group. And they were able to do laminin 332 gene therapy using the LAMB3 gene in the same way that we did it with type 7 collagen. And then they were able to demonstrate expression of laminin 332 in the basement membrane by these same kind of green lines that you saw before. This is called immunofluorescence microscopy. And they showed good expression of the collagen 7 over a long period of time, over a year. And then they were able to show, they took just one patient as opposed to the multiple patients we did, but they really grafted a lot on this patient. This patient had some major skin issues where they had an infection and lost a large portion of their skin. And they grafted over 60% of the patient's skin with these skin grafts. And the patient did really well afterwards. So this just shows that these types of epidermal grafting that we're doing, it just shows the full, even though this is one patient, it shows the full potential of what this could do and how this can really have a huge impact on a patient's life. Another variation on this cell-based gene therapy is instead of using keratinocytes, which are the cells that live in the outer layer of the skin, we also are taking fibroblasts, which are cells that live in the inner part of the skin. And it's known that fibroblasts also produce type 7 collagen. And so what we do in these studies, this is in association with this company Fibrocell, which developed this technology, is we take the fibroblasts from a biopsy, we grow them up in the same type of GMP facility. We transfer the collagen 7 gene in, again, using a viral, this time a lentiviral gene delivery. And then after the expansion, we inject these back into the patient. So these patients, we've treated 5 patients so far, and we're injecting between 1 and 2 wounds, and we inject between 40 and 60 injections total per patient. And we look, again, the same kind of parameters, wound healing and collagen 7 expression. And these are 4 patients. We just treated our 5th patient earlier this month. Again, all patients with severe RDEB. And you can see here, we're able to show collagen 7 expression over time, and we're also able to show some positive wound healing results. So as it stands on this trial, we were able to show that the therapy is well tolerated, the safety profile's good, and we're looking to continue to enroll patients, both pediatric and adult. And if anyone's interested in learning more about this trial, please feel free to contact Kunju Sridhar at this contact information. And then, in addition, so there's lots of really interesting therapies that I'm able to share with you today, and this is another one. And this, first off, I just want to kind of talk about the difference between in vivo and ex vivo gene transfer. So with what we were just talking about before was ex vivo gene transfer, where we take the biopsies from the patient, we grow the cells up in this kind of GMP manufacturing facility, expand them, engineer them with the correct gene, and then deliver them back to the patient. So this It's what has worked for us so far. It's proven, but it is expensive manufacturing these cells in the GMP facilities, and it also involves a virus that goes into the DNA. So there's a slight risk of cancer formation that we have to monitor very carefully. With ex vivo gene therapy, instead of delivering the genes in vivo gene therapy, instead of delivering the genes outside the body, you're delivering the genes right onto the body. So it's simpler. You have a gel that you're just putting on the wound, and in the gel is delivery of the virus of either a oligonucleotide or a viral delivery right onto the wound. So it bypasses the cell manufacturing step. And also, there is no integrative gene insertion, so the risk of cancer is less. So this is one of those therapies that we've already started a clinical trial on and have treated 2 patients, called KB103, that was developed by Crystal Biotech. And this is it's the herpes simplex 1 virus, the virus that produces cold sores, but it's been made replication deficient, and many inflammatory genes have been taken out. And instead, the Crystal folks have put in 2 collagen 7 gene copies. So they're delivering 2 genes per virus. And basically, it does not integrate into the DNA, and it's actually the same kind of a vector was used in a melanoma trial, so it's actually been used in humans before. So this is some work from Jefferson University. This is just an example of a collaborative effort between Jefferson University and Stanford University, where Andrew South there has been able to show that in vitro that skin cells from RDEB patients are very efficiently transduced with this virus to express collagen 7. And then at Stanford, we've extended these studies to be able to show that injected this into a mouse, RDEB mouse skin, you can see really good expression. These little circles are basal membrane around the hair follicles, because mice have a lot of hair. And we also find that you can re-administer it. This is an issue with all kinds of viruses. You want to know, can you repeatedly administer it? And there's at least preclinical data suggests that we can re-administer it efficiently. And we can topically deliver it. So the preclinical studies have indicated that topical delivery is effective. And so that's what the current phase 1 trial is aimed at, is topical delivery of this KB103 to RDEB patients. So we also will do intradermal injection as well as topical application to patient wounds. And healing and safety and collagen 7 expression and anchoring fibril formation is being examined. So this study is open to enrollment, and anybody's interested in this study, please contact Kunju Sridhar at this contact information. And then finally, I just want to mention another in vivo gene therapy. It's a type of a therapy that you put directly on the skin. And this is just, it hasn't begun yet, but it's just initiating now. It's hot off the press. And this is put out by this company called ProQR. And they have this really interesting technology where they have these oligonucleotides, these little chunks of DNA that they put in the skin that will cause the type 7 collagen gene, or the mRNA, to remove one of the exons. Now, an exon is like a chunk, a building block of a transcript that codes for a protein. So type 7 collagens are a really big, one of the biggest proteins, so it's got a lot of exons. It's got 116 exons, which are just little chunks that are building blocks that are put together. What ProQR found is you can remove one of those building blocks, exon 73, and then the collagen still works without that little piece. And it turns out that there's a lot of patients that have mutations in exon 73. And this trial, therefore, is targeting patients or looking for patients that have mutations in exon 73. So this is kind of a specialized, you might say personalized type of genetic therapy. But for patients that do have an exon 73 mutation, this is something that potentially could be a very elegant way to treat their disease. It's very early. It hasn't been initiated yet. I should say also that for both the fibroblast-mediated therapy and this therapy, not only is Stanford enrolling, but also Anna Bruckner, our previous speaker over in Colorado. She's also an investigator in enrolling patients for both this trial as well as the FibroCell trial. And it's going to be also going on in Europe as well. So it's going to be an international trial. And they'll be followed over the long term for this very same things that we've talked about in the other trials: wound healing, safety, collagen 7 expression, and molecular correction. And so anybody interested can either talk to Anna Bruckner, or we can talk to Kunju Sridhar at this number. And I'd just like to, again, thank all of the folks that participated in this. It's been a team effort, and it's been a really fun team effort working with the group at Stanford and all of our other collaborators. And it's also a lot of, it's really encouraging to see a lot of companies wanting to get involved in this orphan disease. You know, it used to be they didn't want to get involved in orphan diseases, but things have changed from a business standpoint to make orphan diseases more attractive to companies. And I'm just so grateful for all of these folks who are taking their time and energies, these different companies and also these patient organizations, DEBRA and EBMRF and EBRP, who are all working together to try to bring these therapies to you. And then if anybody's interested, I'm happy to, if you could email me or call me on my cell phone, I'm happy to talk to you about any of these trials. So I'll close at this point and welcome your questions. Thank you. Okay.
Just scroll down.
Okay. I better get going here. There's a lot of questions. So, the first question: what's the maximum area you can treat with the corrected skin grafts? We can treat 6 grafts the size of about an index card or a playing card. How long does it take to grow the corrected cells and create the grafts? For the epidermal graft, it takes about a month, a little bit less than a month, about 4 weeks. Is CRISPR a form of gene therapy? If so, can you talk about recent positive negatives of that research? Yes, CRISPR has been used preclinically to show that it can induce gene correction. CRISPR's got the advantage in that it can do what we call gene editing, where it can actually change individual base pairs of DNA. What we're doing here in these trials that I described is we're doing more like a wild type replacement therapy. So we're just trying to put in a wild type gene that is just to cover up the defective gene. It's kind of like a redneck living room. You know, you have like the broken TV set, which is like the broken collagen 7, but then you just bring in a brand new TV set, collagen 7 on top of the old one, and you can still watch TV. That's kind of, this approach is like taking a TV repairman and going in there and fixing the TV. That's what CRISPR does. Next question: are there any studies to support that the stem cells of a sibling who is a half carrier of EB but is normal help to treat this sibling who is an active carrier? You know, there is a therapy that is, it's like that, that called the bone marrow replacement therapy, or bone marrow therapy, that Jacob Toller has spoken about, and I believe he will, he'll be able to provide more information about that type of a therapy. But that's a way to be able to utilize the donation of a sibling to help a patient with active EB. Next question, how optimistic are you about the application and full potential of the Italian-German example within the work being done here in the United States? Yeah, I think that, that example is just mind-blowing, the being able to treat such a large area on a patient. And it's not without high cost, it's expensive, but the challenge will be trying to bring the cost down. And, it's the type of a therapy that requires, specialized centers to be able to deliver. So that's also an important factor to consider as well, is development of these specialized facilities to produce the grafts. So it takes a lot of teamwork and effort to be able to get it to that level. But I mean, that example really speaks for itself as to what kind of impact it can have on a patient. If a certain gene therapy treatment ends up succeeding for one gene, like keratin, how easily could that translate to another gene, like lectin? That's a great question. And that's one of the advantages of the work that we're doing, is that it's possible that you could use the same proof of concept to transfer other genes. Now with the keratins, for certain patients with recessive EBS, where there's a lack of a keratin, this type of an approach would work. For the dominant mutations, it's more difficult. You would need something like the CRISPR to be able to correct the dominant mutation. Dominant mutations in general are a little bit milder, at least in the dystrophic area, but they're much more difficult to attack from a therapy standpoint. What is more promising and receiving more investment? Manufacturing skin grafts approach, injecting fibroblasts, gene transfer in vivo, ex vivo, etc. I would say they're all promising. I would like to see them all succeed. I think we have to do all we can to be able to get these patients to these therapies to the patients. For the different studies in progress shared in the slide set, how can we get that info later? Will the slides be shared? Some of what I showed you is published work, and for that published work, the citations are there on the slides. Some of it is not published, and so we can't really distribute hard copies of unpublished data, because then we won't be able to get it published later, and that's important in academics. It's like publish or perish, so we have to be able to publish the data. But we want to be able to get the data published soon. If you don't live close to Stanford, there are other places doing these studies. Well, there's 2 of those studies in Colorado that Anna Bruckner is working with, ProQR and the FiberCell trial. Although we do have patients travel longer distances to Stanford to participate in our trials as well. And we cover the costs of transportation and housing and food while the patients are here. My son has junctional but doesn't produce collagen 17. Will this type of therapy be easy and quick to convert to collagen 17 if it works? Yes, actually this is a group of patients with junctional EB. We used to call it generalized atrophic benign epidermolysis bullosa. And yes, this type of therapy would be amenable to the same type of gene transfer delivery, just like in type 7 collagen. And the type 17 collagen has the advantage of it's smaller, so easier to work with. Are these treatments only available in the United States? Currently, yes, but the hope is that we'll be able to extend these therapies outside the US and worldwide, so all the EB patients can benefit from them. My daughter has RDEB, but it's not severe. We've been approached about the FiberCell study, but are concerned about the cancer risk for the study. Could you speak into that? Well, for these types of integrated viral deliveries, that would be the FiberCell, the Abeona, and then that group in Italy, Germany, who did the junctional EB therapy. This all involves integrating retrovirus. So theoretically, the virus could insert into the chromosome near an area of a gene that could regulate cancer, like an oncogene or a tumor suppressor gene. And that theoretically could activate cancer. It was actually shown to activate cancer in patients who had severe combined immunodeficiency syndrome in France. They treated those patients with a retroviral vector, and many of the patients got better, but some of the patients developed leukemia. The reason why we think it's safer working with this type of a gene delivery system in the skin is that if we see a cancer develop, we'll be able to easily, we'll look very carefully for it, and we'll have a better chance of being able to excise the cancers before they have a chance to fully develop. So that's the reason why the FDA approved these studies, because of that added safety feature. Are there any trials for epidermolysis bullosa aeruginosa? Well, I think there are several anti-itch gene molecular therapy trials. It's not gene therapy, but it is. There's these several trials that have anti-inflammatory therapies that can help to treat itching. I mean, it's hard. It's very difficult problem of EB pruriginosa. We're tempted to use steroids, but the using steroids chronically can cause the skin to thin and create more problems. So it's great that there are some therapies out there that are addressing the itch, and hopefully we'll be able to see more in the future. Last question. A recent article showed that CRISPR can damage DNA even after successful editing. Will we be able to better control it in the near future? Yes, I mean, that's why CRISPR is in the preclinical stage. It's not in the clinics for EB therapies yet, because there's a few bugs that need to be worked out. But the hope is that the CRISPR editing, perhaps combined with IPS or an advanced type of stem cell therapy, can be used as a way to more refine these types of cell-based therapies in the future. And any other questions anybody has? Oh, yes.
So I'm wondering if there are other conditions similar to EB or with similar work, or maybe more common and better funded, or we could piggyback on that would work for EB as well? Are you aware of that?
Could you repeat the question again?
So I'm wondering if there are other conditions that are maybe more common, working with similar work, would maybe better, more funded, we could piggyback on.
Okay. In a way, I think it's more likely going to go the other way. Oh, the question was, are there other diseases that already have genetic therapies or kind of molecular therapies that then EB can emulate and then copy and use for our group of EB patients? And it's actually the case that all these EB therapies are kind of at the cutting edge of skin-based therapy, and it's likely that other diseases might be able to benefit from the EB. That's a good question, though. All right, well, anyway, I'll be around later today. I'm glad to talk to all of you further about this, so I look forward to it. Take care.